Within the PoshBee project, one objective of the work package 9 under the responsibility of partners BioPark and CNRS is to propose an innovative approach, kind of tool, to monitor bee health and to integrate information into the health card. This approach of mass spectrometry is referred to MALDI BeeTyping and is delivering a molecular mass fingerprint from an individual haemolymph sample, haemolymph being the equivalent of the vertebrate blood. In fact, as for the human blood, haemolymph is a mirror of the bee health, and more precisely of their immune status. Through MALDI BeeTyping, we will transform molecular mass fingerprint in a readable report on the bee health for the different stakeholders involved in bee health monitoring. From haemolymph collection, we have a 5-step experimental workflow. First, the haemolymph collection. Second, the sample preparation for mass spectrometry analysis. Third, the data acquisition for the data processing. And finally, the transcription of the mass spectrometry data in a readable report based on a color code like the street light corresponding to the bee health status of the bees. This is what you will discover through this video prepared by the BioPark Karim and Sébastien and CNRS partner, including the consultant Michel, and the help of the L3 and master students involved in PoshBee. Thank you.
The first step of the MALDI BeeTyping is to collect haemolymph from live bees under microscope using a glass capillary and then transfer it to the bottom of the tube. The haemolymph samples are then centrifuged and diluted in acidified water before being spotted on a MALDI sample plate. Once spotted, the samples are dried under mild vacuum. During the drying process, we prepare the MALDI matrix. The matrix is deposited on the spots according to a standard dry droplet procedure, and the plate is dried again. The mass fingerprints profiles MFPs of haemolymph samples are required for each spot on the MALDI plate using the Flex Control software. For data interpretation, the mass fingerprints of haemolymph are submitted to ClinProTools software to statistically compare haemolymph from healthy bees and bees exposed to stressors such as environment, chemicals, nutrients, and pathogens. An additional dimension of the MALDI BeeTyping strategy is to extract peak lists from MFPs using Flex Analysis software for further analysis using R software. The MFPs are used to generate reference spectra for each stressor investigated in the project. This will allow us to score a stressor impact on bee health according to a color code: high in green, medium in orange, and low or no in red. To conclude, the MALDI BeeTyping will represent a building block to the bee health card to assist the stakeholders or the beekeepers in evaluating the health status of their colonies.